I'm Kevin. Thank you all for coming. Oh, okay, so we're recording. I'm Kevin. Thank you all for coming. Today, I'm going to introduce Dr. Gabe Foley. So Gabe is a bioinformatics postdoc working in the labs of Michael Godin and Gary Schenk at the School of Chemistry and Molecular Biosciences at the University of Queensland in Brisbane, Australia. His current research is focused on using ancestral sequence reconstruction to optimize enzymes for biofuel production. He is a Bachelor of Biotechnology with honors, majoring in bioinformatics, and finished his PhD earlier this year under the primary supervision of Michael Godin. His thesis was focused on His thesis was focused on a method for ancestral sequence reconstruction that can handle large amounts of data, and it is this method that will be the focus of his talk today. Gabe has asked that if you have clarifying questions to raise your hand and put them in the chat, and we'll submit them right away. And otherwise, we'll hold bigger questions until the end. So with that, please join me in welcoming Dr. Gabe Foley.
Great, thanks, Kevin. Thanks for the introduction. G'day, everyone. So I just wanted to start by saying thank you to the organizers of the seminar series. So Kevin and Jody, who I've interacted with, but then everyone else who's putting this on, for inviting me to speak today. It's been a really interesting, really engaging seminar series from the ones I've been able to see so far. So I really appreciate the opportunity to come and speak. And before I get started, I'd just like to acknowledge the traditional owners of the land on which I'm speaking to you from today. So for me, this is the Yagara and the Turrbal people. And I'd just like to acknowledge and pay my respects to their ancestors and their descendants who continue to have cultural and spiritual connections to country. And to point out that I recognize their valuable contributions to Australia and indeed to international society. So in today's talk, I'm going to talk about ancestral sequence reconstruction. And I'd like to start by explaining exactly what ASR is, and then importantly, talk about what we can use ASR for. So what kinds of questions ASR enables us to answer. Then I'd like to move to talking about graphical representation of ancestral sequence predictions, or GRASP. And this is a tool for performing ASR that I was involved in developing throughout my PhD. I'd like to pull out a few of the kind of key aspects that help understand GRASP, which are the data structure that we use within GRASP, so the partial order graph, why this is useful, and then to give a high-level overview of how we perform character inference and insertion and deletion inference within GRASP. And then in the last part of the talk, I'd just like to run through some steps of how we validated GRASP. So GRASP is a tool that can use very large amounts of data and complex data. So I'd like to explain exactly what I mean by both of these terms, and then step through some validation slides that show what the advantages of using a tool like this in this context have, what those advantages are for your research. So firstly, what is ancestral sequence reconstruction? So ASR is a method whereby we're using the information that exists in modern day biological sequences. And given the audience and given my own research, I'm just going to be talking about protein sequences today, but it works equally well for any kind of biological sequence, or indeed any kind of sequence where we can model the changes between character states. And we use the information that's in these modern day sequences to predict what their ancestors look like. And now this isn't just theoretical. Once we have this information, we can then take the ancestral sequences into the lab, we can synthesize them, and then we can study them alongside modern day proteins. What kinds of questions can we answer with ASR? So for the next few slides, I'd just like to highlight a couple of the key areas that people are interested in and people, commonly perform when they're doing ASR research. So the first is this idea about discovering key functional residues. So if we imagine for a moment that we have this hypothetical homologous protein family here, and we have some proteins down here that are performing some kind of hypothetical green function. We have other extant proteins that are performing an additional hypothetical orange function. Well, we might be interested in working out what are actually the key residues that are responsible for this shift in function. Now, of course, we could do this just by examining the modern day sequences. So we could look at all of the set that are performing one function and look at what's similar and what's different compared to the other set that performs the other function. But of course, as we know, as proteins are evolving, they're diverging away from each other. They're picking up a whole lot of other mutations that are unrelated to this actual function, unrelated to the change in function. And so to do this on the modern day proteins, you get a lot of background noise that makes this a really arduous task. What we can do instead is we can employ ASR. And so if we perform ASR and we pick up some ancestors, we can take these into the lab, we can functionally characterize them. And if we're able to identify where on the tree the shift in function is actually occurring, then if we think about what we've identified, this means that along this branch, whatever the mutations and substitutions are along this branch, either them or a subset of them must be responsible for this change in function. So what we're doing is we're drastically reducing the search space that we have to consider when trying to find these residues. We don't just have to think about it from a residue point of view either. We can use the ancestors themselves. And so the reason that this works so nicely is that we often see that ancestors perform similar functions to the modern day proteins, but with different properties. And so these are properties that are often very attractive for industry or for drug or pharmaceutical manufacturing. So an example of this, here we have a cytochrome P450 reconstruction that was performed using GRASP. And we can see that the ancestor in orange is far more thermostable than any of the modern day proteins. I should point out this isn't something that's just true of GRASP. This is kind of a commonality across a lot of ancestral research and ancestral reconstruction tools, that what we see is what we recover are proteins that are functional in similar ways, but have different properties like different thermostabilities, different substrate profiles, or different kinetic parameters. And if we combine these two ideas, so the idea that we're creating potentially more thermostable proteins, and that we're also identifying the kind of key residues that might be important for the actual function of this protein. If we combine these ideas, then it's no real surprise that we can also use these ancestors as templates themselves for further rounds of engineering. So either through directed evolution or other protein engineering methods, what we've got now is a really malleable template that works really well. And what we're doing here is we're being informed by the information that's present in the phylogenetic tree, but now we're actually sidestepping and incorporating changes that we don't see in the extant proteins. But the thought here is that we're still navigating within a reasonable part of sequence space, so that we're still predicting foldable, workable proteins. And so all of these examples have kind of been more specific to engineering concerns or more about discovering function. But I wanted to point out that we don't actually need to, and I guess they all kind of assume that you're going to take your protein into the lab and that you're going to actually synthesize it, but we don't actually need to do this in order to gain a lot from ancestral research. And in fact, even just doing this computationally and getting all of our predictions for all of our ancestral points gives you a huge amount of really useful information about how these protein families are evolving. So I'd now like to move to talking about GRASP. And so Grasp is a tool. It's available on the command line for running batch jobs. It's up on Docker, but you can run it as Docker. But we also have a web interface, so a graphical user interface that's up on a web server. And I just wanted to highlight some of the advantages of using the web server, because what we've really tried to do is to make it so that once you've completed a successful reconstruction in Grasp, the web server itself is useful for further exploration of this data. So what we're looking at here are essentially a split screen of the view that you get back from Grasp once you've performed the reconstruction. So here we have the phylogenetic tree, and this is interactive, so you can expand it out and zoom in on certain nodes in the tree. But what the real level of interaction is that you can right click on any of these internal nodes. You can trigger new reconstructions within the tree, or you can view all of the ancestral information piece by piece from all of these internal nodes. So what I mean by that is, if we have a look over here, here we're viewing, so this is the multiple sequence alignment, but then within Grasp, what you can do is you can stack ancestors. So you can choose the ancestors that you want to have displayed. And now we can see we're looking at the N0 ancestor, we're looking at the N1 ancestor, and we can actually just very easily view the differences between them in terms of their substitution differences or their differences in length due to insertion and deletion events. And one thing I should point out is that, so what you're getting back from Grasp, you may have noticed, is not a linear sequence, but it is actually a graph. So this is the graphical representation aspect of the name of Grasp. And although you can get these back as a linear sequence if this is what you're interested in, you can also visualize them within the web server, and you can visualize them just as dot files within the command line version, as graphs that kind of represent a bit more of ambiguity. That's really useful for when we're not, for when we want to display alternative histories, but also really useful for creating more alternatives for doing, for engineering proteins and engineering ancestors. So I'd like to just go a little bit more into the graph data structure to explain exactly the type of graph that we're using. So within Grasp, we use partial order graphs, and these are directed acyclic graphs, and they're really good and commonly used to represent variability in alignments. So they're called partial order graphs because between nodes in the graph, you can define an ordering, but you don't necessarily have to define an ordering. So we can see, for example, this tryptophan in light blue. This does have an ordering in regards to the arginine here, because we see this edge that leaves it and enters the arginine. So we know that it's always going to come before the arginine, but we don't see any edges that are defined, for example, between the tryptophan and this 3 run of asparagines. And so this means that we're not defining a particular order in whether this tryptophan has to come first or second. We can see that when we look at this 2 different multiple sequence alignments that have these 2 different configurations that I'm talking about, but we can actually represent all of that variability within the one graph. Now, I mentioned these are commonly used for alignment, and this is because they're really useful in this way, being able to represent variable regions where you don't actually have enough information to say something definitive about the ordering. They're also really useful because it allows us to delay the calling of an event as an insertion or a deletion. Storing it as an insertion or a deletion, we can delay this until we've considered more of the phylogenetic context. We don't perform alignment within Grasp, so we're not using partial order graphs to do alignment, although you can use a partial order graph aligner as your input to Grasp. But we're agnostic to the type of alignment tool you use. So you can use whatever your favorite multiple sequence alignment tool is. And then we're going to take that multiple sequence alignment, so the traditional way that people would be familiar with, and map that onto the graph structure. And the reason that we map this onto a graph structure and the variability that we're kind of taking advantage of here is the fact that we can also use these graphs to represent ambiguity or variability in ancestors in terms of the potential insertion and deletion events. So we can then consider every possible pathway through a graph as being a potentially different ancestor. So for example, we could consider taking this path along here that includes the LSGE content, or anywhere where we see an edge stepping over nodes, we can consider this as an alternative ancestor that, the way to conceptualize this is this would be a deletion of this content here. And so we've found this to be a really useful way to be able to model this variability and this variation within ancestors. To unpack a little bit more under the hood for the stages of GRASP, what we do is we infer the insertion and deletion history. So this is a history of all indel events across the tree. And so this is going to tell you exactly which parts of the sequence should or shouldn't have content. And this is useful because then we do the inference of the character state. So inferring what amino acids actually should be there. You obviously only doing this on parts of the protein that are actually predicted to be there. And then within GRASP, we're performing maximum likelihood estimation. And so like other ASR tools, we define the two kind of common ways of doing this for ASR. So this would be a joint reconstruction, where what we're getting back is the most probable character for each of the internal nodes. So I'm highlighting it here. We have N7, N8, and N9, and we can see here's the most probable character for all of these nodes, but actually you get this for all of the internal nodes if you're performing a joint reconstruction. The other type of reconstruction that's commonly used is a marginal reconstruction, and here a user has to actually specify which node they're interested in, so you don't get all of the nodes in the tree. But what you do, if we have a look at this diagram, what you do get is you actually get a probability distribution over all of the potential character states at your nominated node. So I'll talk a little bit more about just the distinction between the joint and marginal and how they relate to each other. But then once we have those two bits of information, essentially we just have to piece it all together and create all of these valid paths in terms of indel variants that I was speaking about on the previous slide. And so this is really just drawing all of these potential edges onto the graph so that we can represent this and summarize all this. So I'd like to talk a little bit more in detail about these stages. I'm going to do them slightly out of order. So I'm just going to talk about inference of character state first. The reason I'm going to do this is because we do this in a standard way in GRASP, I'm going to run through this pretty quickly. And then I'll spend a little bit more time talking about the novel aspects of GRASP, which is how we actually infer the indels and how we piece them together. So this is character inference within GRASP, and I know it. So as I said, I'll run through it pretty quickly, but what I wanted to show was just a very simple example so that we could kind of conceptualize and understand exactly what's going on, and just point out, that it's nothing magic. It's very understandable. So what we're asking really is, what's the probability of our internal nodes, so all the possible character assignments to our internal nodes, given what we see at the extant sequences. So in this example, given that we see that sequence 1 is N, sequence 2 is V, and sequence 3 is A. So yeah, using a very small tree and using a amino acid alphabet where we only have 3 possible characters. And this is proportional to asking what's the probability of N0 multiplied by the probability of N1 given what we see at N0, multiplied by the probability of seq1 given the fact that we know that seq1 is N given what we see at N1, that seq2 is V given what we see at N1, and that seq3 is A given what we see at N0. Within GRASP, we do this, so we use a Bayesian network to represent the phylogenetic tree. We're not actually performing Bayesian inference, we're still performing maximum likelihood, but it just allows us to be really efficient in the way that we do this. And so you can imagine that all of these nodes here, if we highlight just on one of the kind of very simple examples, these are really just conditional probability tables where in this instance, we observe that sequence 2 is V. So we just have a conditional probability for all of the possible states that N1 could take. And we can just, get these likelihoods from our substitution model and from the branch length. So from how, from evolutionary time. So the other simplification here in this tree is that I'm considering there's no effective branch lengths. They're all at the same point. They're all at the same evolutionary time. So just to step through this to kind of really make it crystal clear what I'm talking about, if we consider doing this just for one particular internal configuration, so where say N0 is A and N1 is N. So if we imagine stepping through this, then what we're doing is we're grabbing the probability that N0 is A, and here I'm just saying that they're all equally likely to be present. Normally, this is taken from equilibrium frequencies within substitution models. So you kind of, your background distribution for how likely an amino acid is. And then we just multiply this through using the information in these conditional probability tables. So the probability that N1 is N, given that N0 is A, we have, an entry for this, and then we go so on and so forth. That seq1 is N, given that N1 is N, seq2 is V, given N1 is N, and that seq3 is A, given that in this example, N0 is A. So we multiply these all together, and this gives us one of the rows in our joint probability table. And we just have to do this for all of the possible internal configurations. And this is going to let us know what the most probable assignment to these internal nodes actually is. Now, in reality, we don't actually calculate these really massive, because you can imagine these get very, very big very quickly, these massive joint probability tables. Given that we're working within a Bayesian network, we can use variable elimination to speed this up a lot. But I think it's useful to kind of put these out on a simple example just to kind of show what's going on and what conceptually we're doing when we perform this type of reconstruction. The other thing it helps is thinking about the relationship between marginal and joint reconstruction. So now if we wanted to perform marginal reconstruction, as I've mentioned, this has to be centered at a particular node. So if we're interested at N0, then all this is saying for ASR research is, well, we just recognize that there's, multiple ways to get A at N0. So we can just marginalize these. We can sum over the other variables, and we can get an entry just for A at N0. We can do this for all of the other amino acids in at this position, and this is how we get these marginal probabilities. So this explains why they're centered just to a particular node, but that you get these full probability distributions back. So not to belabor the point, but I just wanted to, this is saying the same thing as the previous slide, but it just speaks a bit more about the possible selection space that we have to choose from. Within joint reconstruction, what we're considering in the larger red dashed box is all the possible internal configurations. And then we choose in the smaller dashed blue box, one of these possible configurations. Within marginal reconstruction, so as I've mentioned, these have to be centered at a particular node. So we're actually looking at two different independent reconstructions here. So at N0 and at N1. So if, for example, we're looking at the one at N1, the possible space that we're considering, and here we're just talking about a single position in the alignment, the possible space that we're considering is all the amino acids that go there. And then, so this is the red dash circle. And then what we're choosing is one of these amino acids, so in the blue dash box. So again, this is just to highlight these kind of key differences between marginal and joint reconstruction. To move on to how we perform Indel inference within GRASP, because this is something that's a bit more novel and a bit more interesting, I guess. So within GRASP, we break up the idea about inferring indels into encoding your insertions and deletions. So given your multiple sequence alignment, and then inferring them using either parsimony or maximum likelihood. So we have three methods via which you can encode indels, so position specific, simple indel coding, which is an implementation of a method suggested by Simmons and Okatanara. And then we have the novel method that we implement in GRASP, which is bidirectional edge encoding. So we have these three. Turn the lights back on. So we have these three methods for encoding, and then we have you can either infer using parsimony or maximum likelihood. So you get combining all of these, we have 6 different methods for performing indel inference. So to unpack these a little bit more, position-specific encoding is essentially the kind of simplest way that you could think to do this, right? So it just looks at columns independently and looks at the patterns of gaps that occur there at columns. Now, this is nice because it has analogs with how we do character inference within ASR, which is also column independent. But of course, we know that this isn't really a very accurate model of how insertions and deletions are actually being incorporated throughout evolution. So to say that there's no relationship between contiguous positions or parts of insertions or deletions when we know that insertions and deletions can be multiple characters, it really kind of falls down, but it's perhaps a useful first step, first way of modeling this. So simple indel coding really does try to address this idea, this fact that really we should be considering insertions and deletions as small units rather than just single column events. And so here in simple indel coding, what we're going to do is we're going to classify all of our deletion events in our original multiple sequence alignment, and we're going to call something the same if it starts at the same position and it ends at the same position. So we can see here, that's why we have these this blue deletion in sequence 1 and 3 that get classed as the same, but this yellow deletion, even though it shares a lot of the same characters, it doesn't end at the same position, so it's classed as a totally different deletion event. And I guess this kind of level of overlap leads to one of the big issues with simple indel coding, which is that it doesn't really have a way of handling when we have deletion events that are overlapping each other. So it's going to, so we're looking at slightly different data here than the examples on this side, but it's still the same concept. And we're looking, and what we see is that even though these are all overlapping events, we're being forced to consider them as totally independent deletion events. And so to conceptualize what this means for when we perform inference, this means that we have to perform inference independently for each of these different events. And so the way that I like to think about this is that what we're doing is we're trying to find support for the fact that a deletion event can be placed somewhere in a tree. So we start with the assumption that sequence is there, and then we're trying to find support to place a deletion event. And so this leads us to an obvious problem, right? When we have deletion events that look like they might be referring to the same event, but have to be independently assessed because of this encoding method, that we consider these all independently. And because of this, none of them actually have the support to actually get propagated up the tree. And so this leads us to kind of scenarios that we can reason are pretty illogical, whereby we don't have support for any of the individual deletion events. So we actually end up predicting or inferring that there should be sequence content there, when what we could reason was that actually these are probably more likely overlapping composite deletion events that are going on. I do want to point out that this isn't, so whilst this is the criticism of simple indel coding itself, it's by no means a criticism of the authors of simple indel coding. We're very much aware of this as an issue with simple indel coding. And so actually came up with complex indel coding within the same paper. And then this was also expanded by Mueller a few years later to give us modified complex indel coding. And both of these are methods whereby they recognize that actually there is more of a relationship that exists between overlapping gaps. And so they calculate a step matrix that has edit distance between gaps implemented within it. The problem with this is this doesn't scale well at all, especially to the types of data sizes that we're looking at within GRASP. So we were forced to kind of consider alternative ways to address this problem of how do we associate deletion events that actually appear to have more of a relationship with each other, rather than discretizing them and considering them separately and ending up with some potentially bizarre scenarios. So this is what led us to the third method for encoding indels within GRASP and the novel method that we've implemented. So this is bidirectional edge encoding. And it starts out pretty similarly to simple indel coding. We're working on a graph now, so we're working on edges, but we can still kind of conceptualize this as the same thing as if we were working on an alignment. So we have, for example, this E4 edge in yellow that starts at I and ends at Q, and we can see that this is equivalent to this edge, to this deletion that's identified within simple indel coding. But where they start to differ is that we don't consider then edges independently when we do this inference of where the pattern of gaps should be in the tree. We actually consider this based on considering the set of edges that are either leaving a node, so all of the edges that leave node 0 here, for example, being E4, E3, and E2, and we can see that in this diagram here, or the set of edges as they enter a node. And so because of overhang, the set of edges leaving and entering is not guaranteed and often, is actually very unlikely to be the same set of edges. But by considering them together in this context, it means that we can get away from these issues of not having enough support to propagate edges up a tree, whilst still considering deletion events with the other potential deletion events that are originating or ending at the same positions. So if we think about this, so we perform inference, so either maximum parsimony or maximum likelihood for all of the edges. We have to do this twice, once in the forward direction for each edge, and then once in the backwards direction. So if we look at all of the edges that are leaving this node here, and then we look at the backwards direction for all of these edges. So this means sometimes it's a single edge that is coming backwards from here. So in the case of leaving from this G node or leaving from the L node, this is a single edge. And so we still perform a parsimony analysis or a maximum likelihood analysis to work out where that edge should be placed. But oftentimes this can be multiple edges, and they can be edges that are not necessarily all present in the opposite direction. So this is kind of the additional complexity that we're considering by doing this on a graph-based structure. You're able to think about this in terms of edges, but then think about edges in terms of the context of the other edges that they're interacting with. And I'd now like to just kind of give a brief example of how we patch this together, like how this actually gets represented as support in graphs for graphs, because I think this will help to explain and to understand how you can interpret the graphs that you get out at the end. So what I want to talk about here is the steps I was mentioning earlier, where we have an edge and we go through the tree. And for each edge, we record the support for it at every ancestral point, either in the forwards or the backwards direction. What we're actually doing, and again, here on a small example, we can see it exactly, is we're filling out one of these matrices for all of the ancestors. So this is the matrix specifically for this ancestor, the N1 ancestor here. And then the way to read these is that anything above the main diagonal is going to be our support in the forward direction, and anything below the main diagonal is going to be our support in the backwards direction. And we just record a 1 if it has support for there. So in this example, I'm just talking about parsimony analysis, but we can, so with this N1 ancestor, I've deliberately left off some of the edges just so I can really highlight exactly what the benefit of doing it in this way is, and how you can then gain different levels of support for different paths through a graph. So in terms of reading this matrix, so in the forward direction, so now we have an edge that goes forward from node 4 to node 5, and we can see that this is parsimonious. And so we can see this on the graph now, that we have this edge that reaches forward between these two nodes, and it has this arrow in the forward direction. Likewise, we can see that this edge in the backwards direction, so from 5 backwards to 4, and this green square here is also parsimonious in this direction. So in grasp, we represent something that's parsimonious in both directions as a solid edge. It doesn't. We can also represent the alternative case. So here, forwards direction from 2 to 5, we have that this is parsimonious. So this blue square in the matrix, and we can see that this is why we have this arrow reaching from position 2 to 5 in the graph. But then if we consider the backwards direction from 5 to 2, we can see that this isn't parsimonious for this edge at this particular ancestor. So we represent it as a 0 in the matrix. We don't have an arrow going in this direction. And in grasp, we represent unidirectional edges just as a dashed line to discriminate between them. So there, just to fill in the other ways that we can reach forwards from this node, from the second node here. So going in the forward direction from 2, we can either go to 3 or we can go to 4, and we can see from these purple squares here that's parsimonious in both the forward directions there. And we see that again from 2 to 3, we have an arrow that reaches there, and from 2 to 4, we have an arrow. But then we can see again that in the backwards direction, so from 4 to 2, we are parsimonious, so we have this solid line. But from 3 to 2, we've placed a 0 there, so we only have support in the forward direction. And then just so I mentioned, there's 3 levels of support: bidirectional, unidirectional, and then of course we have that we don't have support for an edge there at all. So just to make that super clear, in the forward direction from 1 to 3, we don't have support, nor do we have support in the backward direction 3 to 1. And so we can see on the graph what this means is we just don't draw an edge there. But what these levels of support then allow you to do once you've pieced them all together on the graph is to actually nominate a preferred path through the graph, given by preferring to take paths that have this level of bidirectional support. So here in this graph, we would step through M, A, P, W, not taking these unidirectional edges, but we still represent them because they represent alternative paths that you might like to explore that might be useful for exploring the properties of these proteins. And now just to the last section of my talk today. I mentioned at the start that GRASP is a tool that can use large and complex data. And so we can see here, we're looking at a representative sampling of ASR studies over the years. And these are all studies whereby ancestors weren't just inferred, but they were actually taken into the lab and synthesized and shown to be functional. In blue, we have studies from other groups, and then in red, we have the study within the GRASP paper where we did for validating GRASP. So what we can see if we look on the y-axis, this is the number of original sequences that are placed into this analysis, so placed into your alignment, placed into your phylogenetic tree. And we can see that none of the previous studies are actually even, so this is on the log scale, are actually even getting to 1,000 sequences. But that GRASP is actually an order of magnitude higher than this, and almost at 10,000 sequences. And I just want to point out that this is by no means a hard cap for GRASP. At 10,000, GRASP is actually capable of doing much, much more data than this. But I like to present it in this way because this is the data at which we're confident because we've actually synthesized and shown that these proteins to be functional. Of course, I guess all of this kind of begs the question: do we need to use large data? And do we need to use complex data? And I think this audience would have a bit of an intuition as to kind of what we get when we use a lot of data and what that can really buy us. But I thought it would be useful just to spell it out in terms of ASR research, what the specific advantages are. And so really it's a matter of coverage, right? So we can either think about coverage in terms of sampling around a particular clade, and just increasing the amount of data that we sample there. And so here, in terms of ASR research, what we're really doing is we're building up a better picture of allowable variability at different parts of the protein. We can, as well as thinking about this in terms of sampling from a particular part of the clade, we can also think about including more data as allowing us to include a more breadth of data. So sampling either from distant homologs or additional homologous protein families. And this allows us to infer even more ancient ancestors, to go back even further in time. And this is enabled really greatly by being able to use a lot more data. And then finally, on the matter of complexity, well, we know that proteins don't just evolve through substitution events or mutation events. We know that they're also evolving by gain and loss of little blocks of content, so through insertion and deletion events. So if we have a framework whereby we can model this complexity of insertions and deletions, then this means that we're able to model the underlying biology and the underlying true history in a more realistic manner. Of course, with all of these advantages of large data come challenges. We have to ensure that on the smaller data set sizes, our predictions are still accurate. We have to ensure that once we start using more data than the other tools are able to process, that these predictions remain accurate, that we don't go careening off into some weird part of sequence space. And in terms of Indel events, we have to make sure that we're able to model these, not just so that we can get predictions out of them and we can piece together these graphs, but also that the history of Indel events that is implied by our graphs actually makes sense given what we know about the underlying biology and the evolution of Indels. So in terms of validating grasp, I'd like to just step through essentially these 3 points. And this is what we do in the grasp paper as well. So how does it compare to existing methods on data set sizes that are able to be used by all methods? Once we get beyond these once we get beyond data set sizes that other methods can handle, does it still give valid predictions? And then in terms of indels, not just can we model them and what happens with the different indel methods, although we do talk about this in the grasp paper, but can we actually use insertions and deletions to our advantage and tap into this as a novel form of engineering our ancestors? So on the question of comparability on smaller scales of data, so we took a cytochrome P450 2U1 subfamily. This is 359 sequences. And then we gave the same alignment, the same phylogenetic tree, same substitution model, and we used this to make ancestors with GRASP, and then 2 other ASR tools, FastML and PAML. We picked an ancestor from each of these, and then what we saw is that regardless of the method that we used, all of these ancestors expressed. They're all active towards the expected P450 substrates. They all have similar comparable thermostabilities. So on the question of comparability on small data sets, it certainly seems that GRASP's predictions are in line with what you would get with other tools. And so this was experimental work. So the wet lab work here of characterizing the proteins is performed by Connie Ross. Then when we get to upscaling, so beyond that, which other tools are able to process. So we did this with the DHAD reconstruction. We start with a small tree, although already we're at around 1,600 sequences. We pick 3 ancestors of interest, and then we just upscale this tree by sampling around this phylogenetic space. We get to around 9,000 sequences, and we pick the equivalent nodes from this tree. And then we take 6 ancestors, so 3 from the small and 3 from the large tree, into the lab. And again, it's a similar story. All of the 6 ancestors express, they all look like DHADs. They're all active towards the expected DHAD substrates. And this experimental wet lab work was performed by Scott Bottoms. So again, it certainly seems like even when we start to get to these really much higher orders of magnitude data set scales, we're still getting back valid ancestors that are still workable and still give you foldable, useful protein. And so the last part that I want to talk about in the validation is how we're able to use insertion and deletion events. So I mentioned that it's not just about being able to model them appropriately, but then being able to do something with them once you have this variability expressed as a graph. So what we did in GRASP is we identified parts of the tree, and this is a cytochrome P52U1 subfamily tree. We identified parts of the tree where there was where an insertion was about to occur. So here we can see along node N51, we have this LLSPP insertion that's about to occur. And then we took this as a little block of content, and we actually placed it back into the N2 ancestor where it was not predicted to be present. We did this for a couple of other different lineages, identifying where they're about to occur and then moving them into ancestors where they're not predicted to be. And so we term these ancestors that are hybridized in this way as hybrid ancestors. And really the hypothesis here was that even though we're moving around fairly substantial little blocks of content that you might reasonably expect would disrupt protein folding or cause these to become non-functional, that because we were identifying parts in the protein where we knew insertions were about to occur within a few generations, and because we were placing these back into ancestors that were at a similar evolutionary time, the hypothesis was we'd be dealing with some of the epistatic effects, and so that these ancestors would actually be amenable to accepting these blocks of content, even if they weren't predicted to have them there. So we did this, we reconstructed all of the native predicted forms of these ancestors as well as these hybrid ancestors. And indeed, this is what we saw. So for all of the ancestors that we synthesized and took into the lab, again, this is wet lab work by Connie Ross, we saw that they all worked, they're all active towards P450 substrates, they all look like perfectly functional P450s. But this is, so that was great, but it's kind of not even the most interesting part of this story. And that's that what we actually saw, these little modular blocks of content were actually able to change the functional properties of these ancestors. So here I'm just highlighting a few results. But what we can see, for example, with the thermal stability is that if you have this LLSPP insertion, so regardless of whether or not you're predicted to actually have it, if you either retain that block of content or you have it placed into you, it increases the thermal stability that this ancestor can operate at. And we saw a similar kind of pattern with these insertions opening up and changing the substrate profiles for activity with different P450 substrates. What I should mention here is that we can't really predict ahead of time which way these things are going to go. So there's no, it doesn't necessarily always follow that inserting this content in is always going to raise your thermostability. But really what we're trying to highlight here is that what we're doing is we're opening up more of this sequence space that's now available through using these hybrid ancestors to be able to generate content that we think has a higher chance of actually being able to fold, and that then we can explore the variation in these functional properties on these new hybrid ancestors and these new variants. Great, so that brings me to the end of the talk. So I just wanted to point out exactly where GRASP is available. So the web server is up. You can follow this URL here. I've also included the URL on the information for this seminar series. You can take a photo of this QR code that will take you straight to the web server. But I also just wanted to point out that we do have quite a lot of information surrounding GRASP that's available as part of GRASP Suite. So here you can get to the web application. You can also download the command line tool. You can, and we have a lot of tutorials and guides that talk you all through the process of performing this kind of work, including curating your alignments, identifying what a good or a bad phylogenetic tree is, right through to using GRASP and then interpreting the results that you get out of GRASP at the end. GRASP is open source. So here's a link to the code repository. And then I'm just highlighting a few of the publications. So we have a preprint up on bioRxiv at the moment. And this talks through a lot of the validation steps that I was just talking about, and introduces GRASP as a method. And this is in review at the moment. So we're hoping that this gets published very soon. But then I also wanted to highlight this methods in molecular biology chapter that came out earlier this year. And this is really useful because it explains how to set up your experiments so that you can actually get these insertion deletion mutants so that you can actually make these hybrid ancestors. And this explains it not just from a computational point of view, but also from a wet lab point of view. So what experiments do you need to run after the fact in order to validate these ancestors? This is quite a lot of information. The easiest way to kind of access this and get into the GRASP ecosystem, I'm suggesting, is just to search GRASP ASR, and this should give you back all of the links to the relevant publications, as well as the educational material, as well as the tool itself. So I'd just like to say thank you. So I mentioned this is mostly work that was done within my PhD. So I'd like to say thank you to my PhD supervisors, Mikhail Bowden, Liz Gillum, Ross Barnard, and Michael Landsberg. But then also a massive thanks to all the people who've worked on developing and validating GRASP over the years. So it's a big list of people, but I wanted to specifically highlight the second authors, Ariane Mora, Connie Ross, Scott Bottoms, and Leo Stutzel. So Ariane did a lot of the computational implementation within GRASP and testing within GRASP, and then Connie, Scott, and Leo did a lot of the wet lab validation I've been talking about today. And then finally, just to highlight again, Liz Gillum and Mikhail Bowden, both for the design of experiments within GRASP, and in Mikhail's case, the coding of a lot of the inference engines that GRASP runs on. And I'd like to finally just leave with a take-home message, which is really just summarizing these points in the last part of my talk. GRASP is comparable to other methods on smaller data set sizes if you want to make ancestors, retains valid predictions when you start using much, much larger data, and that these insertions and deletions, as well as being manageable in ancestors, they actually allow us to engineer ancestors in a new way that opens up more of this ancestral sequence space to really being engineered and looked at. So thank you very much for your time, and I'm happy to take questions.
Great. Thank you so much, Gabe. That was a great talk. I think I've added the ability for everyone to unmute themselves. So you guys can either put questions in the chat or unmute yourselves and ask. So I think one person, Jeff, raised their hand. You
want me to just unmute?
Oh, I think participants should be able to unmute themselves. So Jeff, you can go ahead and ask your question.
I'm unmuted. Very interesting work. I was kind of curious about what the exact speed up is. In my experience, like the slower part, or the reason that you're using smaller data sets for ASR, is primarily the phylogenetic inference. But I wasn't 100% clear to me from your talk whether GRASP actually was doing that phylogenetic inference or not.
So no, so GRASP isn't doing the phylogenetic inference. So to use GRASP, you have to provide as input your multiple sequence alignment and your tree already. So all of that phylogenetic inference has to happen outside of GRASP. So yeah, there's no speed ups for that, unfortunately. But we do, even the inference method then, once you have the tree, is drastically improved by using the methods that we do.
Great. I'll ask just one follow-up on the insertion work. Are those insertions primarily located like near the active site, or they just happen to be kind of anywhere in the protein?
Yeah, so for the 2U1 insertions and deletions that I was talking about, so when we model where they actually were, so they actually aren't really near the active site in this case. So they're actually in a couple of loops that actually I have a skip forward just a bit. So they're actually in, so a couple of loops that are actually pretty far away, and they're in pretty disordered regions. So in this case, no, they weren't. I guess so. The issue with this is that in order to generate insertion and deletion variants, you need some level of variability of insertion and deletion around that part, and that at least with a lot of the data we've seen, this happens to be this generally happens away from these like well-conserved parts, because these well-conserved parts are just not going to have variability of insertion and deletion. Although this could be true for other proteins, it's only true for the ones that I've looked at. But certainly in the case of the 2U1s, no, they're actually quite far away, and they're in these the DE loop and the AA loop within this particular protein.
Great, thanks.
Thanks, Jeff.
So we have a question from Chris. You can go ahead and unmute yourself.
Hi there, Gabe. Thanks very much. That was a really fantastic talk. I was interested to understand how GRASP might work within sort of, say, immune system molecules where you don't have a massive number of insertions and deletions, but you have a really high number of polymorphisms, and the sort of complementarity from them is the antigen that they bind.
Yeah, so I've kind of highlighted and spoke a lot about insertions and deletions just because that is one of the novel aspects of GRASP, but it does work. You don't need insertions and deletions to use this tool, and you get a lot out of it not using, you don't have to have insertions and deletions in your data. So I think one of the advantages in terms of being able to use more data is just that question of being able to sample more around there. So I'm not particularly familiar with what the actual challenges would be in using highly polymorphic proteins. But if it's the case of being able to sample more variability, so include more extant data so that you can get a better sampling and a better distribution of the possible variation at each individual site within the protein. But I think that's something that would be like very well suited to using something like GRASP so that you can use a lot more data in your analysis. Does that answer the question?
Yeah, absolutely. No, and it's sort of something that I'm already thinking about how I can play with it. So thank you for making it open source and making a nice web interface for it as well.
Oh, awesome. Yeah, fantastic.
Gabe, I have a question. This is Jody again.
Yeah, hi, Jody.
I've heard of kind of some instances of ancestral sequence reconstruction, showing that a lot of enzymes have lost, first they've evolved new function, and then they've lost thermostability through the years. For instance, like thermophilic bacteria having enzymes that will maybe change their specificity. And then there was previously a paradigm that if you have a new specificity, you might lose thermostability. And some people have, shown in the past that that's actually not the case, that you actually, when you move from a thermophilic to a normal, a bacteria that lives in a normal temperature, you're actually losing thermostability just by the accumulation of mutations. So I'm just really curious if you could comment a little more on just GRASP in general, and if you could see that, helping us understand thermostability, making current proteins more thermostable, et cetera.
Yeah, absolutely. So it's a really cool question. It's one of the really big, interesting questions in ASR, right? So this idea that you get proteins that are more thermostable, and the question of how much of this is tapping into the actual true biology that these are more thermostable, how much of this is how much of this is an artifact of the ancestral reconstruction process itself. So just quickly, again, not to kind of repeat the same answer, but I think that one of the big advantages of GRASP is that we can use more data. So using more data really does allow you to incorporate more variation to increase the robustness of these predictions. So I think it's really going to help in terms of answering some of these questions about thermostability. In terms of things gaining or losing thermostability, so I guess that like there's a few clear examples in the literature where we have gains of thermostability that can't be explained or probably aren't explained just by the fact that these were more thermostable because of increases in the Earth's temperature at the point that we're going back to. The other point that you touched on, that this could be more about becoming specific in terms of your function, I think is potentially a more accurate picture of what's going on, that these proteins were more generalist proteins, and that then, so this is certainly what we see. So certainly within GRASP, within some of the publications that were published over the last few years, that you start off with a more generalist protein and then different clades specialize in different ways, but that there's a trade-off that you have to lose some of this, some of the stability of your protein in order to be able to perform these specific functions. And so you can kind of track that as it comes down different clades. The difficulty here is that we have to be careful when we talk about generalist and specialist, because we have to make sure that we're not just, so what's often done is that we know substrates that our proteins should interact with, and then we make ancestors, and then we test them on this range of substrates, and we see, okay, well, they don't interact with this, or they only interact with one now, so we now class this as a generalist, and we can show that all of the extra ones all have this different array, so here they're specializing out. What we have to be really careful of is that we're not just using confirmation bias in our selection of things that we're actually testing. Because there has been a couple of papers out recently that have shown, well, actually, if you're less biased about the things that you actually test on for your ancestors, you can show that they actually have still pretty high substrate profiles. It's just that they're substrates that are different, substrates that we didn't really weren't really concerned about with the extant proteins. So it's an important question about generalist moving to specialist being indicative or being related to losing stability. But I think we do have to be careful that we make sure that these are truly generalist proteins that we're talking about, as opposed to just proteins that have a different range of activity. So hopefully that, I don't have any kind of definitive answers, because I think it's all very much still open and still up for exploration. But hopefully, yeah, a tool like GRASP does make it easier to kind of start posing and answering these questions in a bit more of a high throughput and high data way.
Thanks. Yeah, that's really insightful. Any other questions? We'll keep it open for another minute or two.
If there is anything that people have at a later date, please feel free to reach out to me. Either through GRASP, you can get my contact details, or via Twitter or anything. I'm more than happy to talk about stuff that might be more specific or related to projects that you're working on.
Okay, let's thank Gabe for an awesome talk, everyone. And we will see you all in 2 weeks for our discussion panel with our past 3 talks. Thanks so much, everyone.
Thanks, everyone.
Thanks, everyone.
Thanks, Gabe.